Hello everyone, welcome to my new video. Today we will study about the secondary growth. It is the part number 1.1 of plant anatomy. What is secondary growth? Secondary growth means increases in diameter. It increases in the girth by secondary meristem and lateral meristem. Here, two types of growth, stellar growth and extrastellar growth. The growth inside the stellar region is stellar growth, and the growth outside the stellar region is extrastellar growth. For stellar growth, we discuss about here is a pericycle. Here we already have an intervesicular cambium in dicot stem or root and intravascular cambium, and we also have the medullary rays. And when the medullary rays de-differentiation, when medullary rays goes under the de-differentiation, it forms secondary meristem, and then secondary meristem form the intervascular cambium, which is half is primary and half is secondary. And then secondary xylem form more and growth in stele rather than phloem. Secondary xylem is form more and it will help in the growth of stele by vascular cambium. And then second part is extrastellar growth, when the cortex, which is made up of parenchyma, is go under the de-differentiation, it form the crocambium, where epidermis rupture and crocambium form. Epidermis is rupture and crocambium take place of the epidermis and growth outside. In brief, how the secondary growth is in stem. Here we have they are medullary rays, they are phloem and xylem, which is secondary intravascular cambium. We already have secondary intravascular cambium and primary medullary rays. We already have secondary intravascular cambium and primary medullary rays. And then primary intravascular cambium, here primary intravascular cambium come, then primary medullary rays de-differentiate and form secondary intravascular cambium. We already have intravascular cambium secondary, and secondary intravascular cambium come from the de-differentiation of primary medullary rays de-differentiation. And then vascular cambium is made a ring, and inside secondary xylem is more than the secondary phloem, so the second degree medullary ray is also formed. And second growth in the cortex region, when central cortex goes under the de-differentiation crocambium or phallogen. The another name of crocambium is phallogen. Secondary degree in origin completely. Secondary in origin completely. Crocambium is secondary degree in origin completely. Second degree cortex, here complementary and crook. Crocambium is also known as phallogen. Center, the secondary cortex and phallodome. Here center, the secondary cortex and phallodome. In center, secondary cortex, which is also known as phallodome, and the periphery complementary, which is living, and crook dead. Let's study about the croc. Croc is the dead which is suberized and impermeable to the water. Complementary thread go under the pressure and lens shaped its form, which is called lenticels, and it help in the gaseous exchange. It only seen in the dicot or stem, and the secondary cortex and complementary cortex and cold. Their collection called peridome. Peridome is formed by the croc, another name is phloem, and crocambium is phallogen, and palindrome is the secondary cortex. These all made the peridome. What is bark? Every tissue which is exterior to the vascular cambium is called bark. Peridome plus second degree phloem is called bark. Secondary growth in root is similar to the stem, but the vascular cambium is completely secondary in origin, where the vascular cambium is half primary and half secondary in root in stem, but here in root in secondary growth the vascular cambium is completely secondary in origin. Pericycle plus conjunctival tissue. How annual rings is formed? It is formed in the woods like a plant which is in temperate region due to the chemical climate fluctuation help to determine the age of the plant. Annual rings, one light ring and one dark ring called 1 years. It helps to determine the age of the plant. And light spring root, summer early root that spring cool high cambial activity in spring. The xylem is very high, the lumen is wide, lighter in the weight, density is decreased, so they are seen light in color. Dark, late, or autumn or winter wood. Winter, the cambium activity is less, xylary element is low, and lumen is narrow, so they are dark in color. And then compare the hotwood and sapwood. Hotwood is also called a duramen, and sapwood is also known as alburum. Secondary xylem is in the center and non-functional. Density is very high and it is very dark. Microbes cannot attract the duramen, which is called hardwood, and no deposition of any organic substances in the sapwood. Secondary xylem is at very and it is functional. The density is very low. It is light in color. Microbes cannot attract. Microbes can attract the sapwood. And there is one interesting thing in the hotwood. There is tylosis formed in their parenchymal. There is a tylose is formed in the hotwood.